Okay. Hi viewers, this is Madhu Biotech channel. Yesterday's class, before class, we have discussed about that isolation of desired gene, cleavage of DNA, number of fragments. Now the next point is insertion of rDNA into suitable vector. So before of that, this is the isolation of DNA is completed. As usually same here also, first of all we want to isolate of vector. So the vector, so at the same way, the first of all we have to treat it with the number of different types of enzymes degradable to that cell walls, and we are isolated pure form of that vector. So this is called also vectors. The vector is mainly used as a plasmids. The plasmids are located inside of the bacterial cells. The plasmid means, the plasmid is the double stranded, the circular extra chromosomal molecular is called also plasmid. So these type of the plasmids we are using as a vector. The vector means the foreign gene, it carries the one organism to another organism. That vector is called also, that is called also vectors. So this is also carriers of vectors. Plasmids is called also carriers of foreign genes. This is the completed structure of rDNA technology process. So first of all, then we want to isolation of this vector with the using of different types of enzymes. We are using degradable to the cell wall, then we are isolated. So here the enzymes, the foreign gene, we are cut into a number of fragments. Here which type of enzymes are we are used, as well as at the same time here also we are using same restriction endonucleases. Why means both of the same endonucleases, it cleave the same type of the fragments. So this RNA, this DNA, the foreign gene inserted into this suitable vector with the help of ligase enzymes. With the help of ligase enzymes, this ligase enzyme, it joining with the bonds of hydrogen bonds foreign gene to vector DNA. So this type of the DNA is called also rDNA. This type of DNA is called also rDNA or cDNA. cDNA means chimeric DNA, recombinant DNA. This is the next step. Then the next step, further another step is that this recombinant vector in vitro, in vivo, amplified number of copies with the help of PCR technique. Then after that, this amplified gene we are inserted into that suitable host cell. But the recombinant DNA, normally they did not inserted into this suitable host cells. The host cell did not acceptable of rDNAs. Why means the bacterial cell, it made up of different types of phospholipids and protein murine type of the particles. So here the DNA particle is that hydrophilic organisms. The bacterial cell wall, it made up of hydrophobic phospholipid particles. Directly did not acceptable to that host cell. Because of this reason, first of all we want to competent to this host cell with help of calcium chloride treatment or micro injection method or so different types of ways. The rDNA, it transfer through host cell. This process is called also transformation. This process is called also transfer. So first of all we want to competent to host with the help of different treatments. The first treatment is calcium chloride treatment, second one is that electroporation, third one is micro injection, and fourth one is that bioelastic method. So already the host cell, it made up of different types of hydrophobic lipids bilayer particle. The DNA particle is hydrophilic. So this is hydrophilic particles, they did not enter into the host cells. Before of that we have to competent to that host with the treatment of CaCl2. So CaCl2 means the host cell, first of all the host cells we are poured in a calcium chloride solution, then the cell wall it solubilized, then this host cell then again we are put into that ice, then that time we are added with rDNAs, so that time the rDNA inserted into this place. Then after that we have to take that heat treatment. First of all calcium chloride solution, then ice plus ice, again heat treatment. So then the rDNA it enter into that host cell. This is the one technique. And another one is that electroporation. The host cells and rDNAs both are mixed and we are poured in a one test tube. The test tube we are put into the one machine, that is that electrophoretic machine. When we are treated with electrophoretic, so directly the rDNA enter into that host cells. That is the second one, electroporation method. Micro injection, most probably this is used for that animal cells. Directly one micro injection present in this place, we are directly take that injection and this is the nucleus, directly inject into that cells. This is the another method. And last one is that ballistic method. This is called also shoot gun method. When the tungsten and gold particles we are covered with that rDNAs and directly shoot that host DNAs. This is one type of method. With the help of this method, the rDNA transfer through vector into host cell. This process is called also transformation. This process is called also transformation. Okay, the suitable host cells, that host cells is called also recombinant host cells. The next step is obtaining desired gene product. The recombinant cells we are using of culturing techniques, we are preparing number of growth rates. So the different types of, we have to amplify that number of copies, we have cloned number of copies of these desired gene products with using of batch culture methods. The small scales we are using of with help of petri dishes, we are culturing of these rDNAs, we are preparing with help of specific medium, we are culturing number of group of the bacterial cells, This is the small scale industry, we are using of these petri dish methods. Here the host cells transferred through the one petri dish, that petri dish it containing that medium is culturing medium, we are inserted into that fresh mediums and we are removing to that old mediums and we are culturing and developing of different types of number of copies of that host cells. Okay, small scale we have to providing that petri dish method, that is the one specific type of culture we are producing number of host organisms. And the large scale industry we are preparing thousands of the cultures into with using of bioreactors. In this culture medium we are added into that specific protein, that is that recombination protein. That protein we are added into these cultures. So this is that one specific rough diagram of bioreactor. With help of this bioreactor we are preparing amplifying of host organisms. So bioreactor means we are providing optimum temperature and humidity and oxygen in the in vivo conditions. We are preparing large scale cultures. This culture is called bark culture, and this is the structure of bioreactor. And the bioreactor it can contain the two stirring type of that jackets, it covered with these jackets, and these jackets is called also cooling jackets. It gives that cooling to that bioreactor inside organisms, and here developing of host organisms inside. So these jackets are giving to the cooling sides, and one spreader also is present in the bottom of the bioreactor. This is the porous type of the plate like of the structure. So this structure it is called sparger, and with the help of these pores, the harvest, when the products are harvesting, is it is contained that the below part, this is the harvest line. And one more center of the part of this bioreactor, it can contain one large stirrer, and the stirrer it can contain number of blades. This stirrer it stirring of these bioreactors. Then when it is stirring off in this bioreactor, the mixing of that medium and that culturing and developing of growing of that number of host bacterium cells. And here some of the lines are providing at this, we are giving that optimum temperature, humidity and oxygen in these reactors, which contain special parts are present. That parts is called also lines. So the lines are here. This is the one line is present on the top of the bioreactor, that is the inoculum line. In this help of this line, we are adding of inoculum inside of that bioreactors. And another line, this is the antifoam agent line. We are adding, with the help of this line, we are adding antifoam agents, we are adding inside of this bioreactors. And another line is present top of the top of the bioreactor, that is nutritional line. With the help of this nutritional line, we are adding into of number of mediums. When the medium is completed, we are adding, and in the bottom of the place we are removing of wastage materials. And another line is present top of the bioreactor. This is the pH. We are maintaining of pH of this medium, and every time we are providing of dissolved oxygen with the help of this line. This is called also dissolved oxygen line. And one temperature sensor and control unit are present, and with the line of this cooling water line, we are entering into the cooling waters. With help of this bioreactor, we are preparing large amount of host organisms. This is the this is called also bioreactor. Then last step is that downstream processing. With we have downstream processing means we are specified identification and purified testing. That testing products are different ways we are testing that products are useful for human welfare or environment conditions. That purified material and purified host organisms, we are secreted number of products, and we are marketing, and we are providing to that marketing. So this is the whole process of rDNA technology. Okay, so thank you one and all of subscribe my channel.